Okay. So this will be the third instruction slash class. Okay. Let us get started. Uh, Let us first discuss the last problem which I had shown. I think most of you are trying to do it. Uh, Anyone who did not manage to do it? Okay, I think most of you solved. So the first part was very easy. So I'm not doing that again. And for the second part, what did you do? C1 versus X to this thing. What was the answer? Transcritical. When C1 is negative, C2 is just to this thing. Okay. Something like this, right? It was a transcritical. Now, how can we think this in terms of physical? Can you tell me what was the value of C1? K1A minus K2B. This K1A minus K2B. Okay. Now, if you think in terms of a physical picture, when C1 is negative, what does it mean? That your K2B is higher than K1A. So the consumption rate of X in the X plus B reaction is much faster compared to the production rate in terms of the K1. So it is being produced very slowly but consumed very fast. So ultimately, after a long time, you won't have any leftover X because it is being consumed way faster than it is being produced. And when C1 is positive, it means that the first reaction is fast. So you have good accumulation in terms of this 2x term, but the consumption portion is happening very slow. So after very long time, both the rates will adjust out and you will have a steady positive concentration. So in the past two classes, we had been discussing who does systems biology, why we need to do systems biology. And after that, we had moved on to some simple problems, how to model biomolecular processes and such. We had discussed some population kinetics model in terms of logistic growth models and predator prey models, and taken a look at some basic bifurcation diagrams. Today, what we'll start to discuss is something which is the most integral part of systems biology, and that is called network motifs. Okay. And this is how currently systems biology, or at least theoretical systems biology, the modeling portion of that is entirely based upon the fundamental principles which will be discussed. Now, I must acknowledge my professor because the slides we'll be using in the discussion today are from him. It was part of the course which he had taken. And this is the lab which I worked in my MSc project. So if you see this one, he is Professor Sandeep Kar, and I worked with him. And these are my other lab mates. It's a pretty good lab. So any one of you, if you want to join either for MSc or postdoc, it's hopefully I can inspire you at least in the at least towards systems knowledge. Okay, let's start it. So this is what we had been discussing in terms of phenomenological modeling. As I mentioned, we had some enzyme kinetics, we did a chemostat problem. And in the last class, we had discussed some bifurcation diagrams on how they help us understand the long-term behavior of certain dynamical systems. We had done the Gray models, harmonic oscillator models, and some theoretical bifurcation diagrams in terms of if we take some model equations. Right? But we had not discussed any particular biology, at least in terms of, suppose in the first class I had shown that it was the immune system, the system, in terms what is the system in biology, the immune system. But till now I haven't discussed any particular sort of biological picture with you, where you could get an idea that how does this particular mathematics of bifurcations help us in terms of biology, or at the cellular level biology or molecular level biology. Today we'll be discussing that. But before discussing that, we need to understand if we want to model the processes in biology, what are the scales which we are operating? So what are the length scales or the dimensions which we need to operate in, and what are the time scale which we need to operate? For example, if you look at the size scale, if you think of the basic or the most common thing which comes to mind, it's human body. Okay? One of the most evolved forms of any biological organism. And if we gradually move towards more and more minute and smaller scales, we can go from mouse, then you move to insects. Definitely, then you move on to bacteria and microorganisms. So ultimately, if you go further down, you're slowly moving into the subcellular levels and ultimately atoms. And if you think in terms of the time scale, if you go in the reverse direction, the fastest processes, which are chemical reactions or transition state, their lifetimes, it is usually femtosecond. So that's why you do like femtosecond spectroscopy, ultra fast spectroscopy, where it's used. And gradually you move upwards to biomolecular interactions, then protein translation, transcription. Then you slowly move into these particular things, which are cellular regulations. So for example, cell cycle. What is the time taken by one cell to divide? You have glycolytic oscillation in terms of you have circadian oscillations, which is the body clock of humans. We operate on a roughly approximately a 24-hour body clock. Right? And if you move even further, you have generation time. How fast do organisms reproduce? Right? And ultimately, you go into the evolutionary time scale, which happens across billions of years through nature. And each particular, especially from the cellular level and upward onwards, is actually a subsection of systems biology. You can do molecular systems biology, you have normals, where you deal with cells, cellular systems biology, and you also have evolutionary systems biology. The processes which we'll be discussing in this course are mostly focused on these green dots, which are at cellular level. So cellular length scales and time scale related to cellular and biomolecular. Okay. So how does a mammalian cell function? If you think carefully, just like the picture I had shown you, every system, in this case, if you think of a mammalian cell, it receives some input signal from the environment, from certain drugs which you had, or from any other sort of chemical signals or physical signals it's found in its environment. It processes it and gives a specific output in terms of a physiological response. So it's essentially an information processing system. Whatever information or signal it receives, it processes it, it gives an output result. This is exactly what a computer does, right? You put a particular input or a signal in terms of either your mouse or your keyboard or whatever prompt you put in. It processes using the processor and in some cases the GPU, and you get a specific output. So does the cell compute exactly like a computer? Or if there are some differences, what are the differences? Particularly, both are information processing systems, so you can compute using them. The major difference, the first thing which comes to mind is electronics are silicon-based systems, whereas life is a carbon-based system, organic-based. What are the other distinctions? There are some basic distinctions which you can think of. Usually computers are solid state transistors, capacitors. Biology mostly deals with either liquid or semi-solid. The most important difference being that in terms of computers, they are usually digital computers, which we are usually working. So they work in binary, ones and zeros, and then they process into further higher level languages. Biology operates in analog. So you have proper what can you say? It's a continuous time process. You don't biology doesn't deal with zeros and ones as such. You have either protein increasing, decreasing. You don't have stepwise 0, 1, 0, 1 sort of expression. You have a continuous expression all throughout that. Then further some other classifications which are possible. Biology is usually self-reproducing, computers are not, definitely. And computers are designed, whereas biology has been evolved by nature. So it's designed by nature through evolution. And computers are of course programmable. You can design how you want them to work. Cells, hardwired by nature, you can't change them to a large extent. You can, but not to a very large extent. So we know that in case of computer, it's a silicon-based system, it's hardware. If we look that how exactly things, or rather information is processed in a computer, it has to go through a circuit system, right? So it's a hardware through circuits. how does the circuit system, if at all, for a cell exist, how does a circuit system in a cell operate? So, in terms of computer, we have transistors, we have these which are silicon based, you have capacitors, you have a CPU, you have a GPU, you have a mother, so everything resides on the motherboard and they communicate through semiconductors. So if you think in terms of electrical engineering, you can make all these complicated diagrams, and if you give it for printing, some manufacturer will give you a motherboard. But how does the circuit of a cell look like? So this is how a circuit of a cell usually looks like. Everything seems sort of crossing over each other, and yeah, so do these processes occur simultaneously or do they not occur? Are they sequential? If we cut down all the complexities, and I tell you to identify some particular patterns, you see there are some recurring patterns in these lists. So if you see the red boxes, you see between any two species, you have double ends. So for example, in this was X in this one, you see this is positive, this is positive. If you see the CDC25 cycle, this is a positive arrow, this is positive. See, it's positive. And you also see some hammerhead. So this is called a hammerhead arrow, and I'll call this a normal arrow. You see between this BCL and BHT, there is a hammerhead arrow pointing in both directions. In the CDH1 cycling B, there is a hammerhead arrow pointing in both directions. So let us establish this. So whatever is the hammerhead is a inhibition or a degradation. Whatever is positive is definitely opposite. Okay. Activation. This is the terminology which will be used. So you see, even though it might seem that there are a lot of complexities in the network, you can identify small patterns within the network which are constantly repeated. So since something is being constantly repeated, it must have some particular function which is ubiquitous, right? It has some function which is so universally needed that nature keeps repeating it across different species, across different molecules, because the output must be well established and robust. We'll take a look at it. So before that, so these what you see, these are something called as network motifs. So what are network motifs? What I showed you, these are patterns of activation and inhibition which act between molecular species. And the biological sort of counterpart, or not a counterpart exactly, in the biological context, these are called modules. Right? But if you think in terms of electrical engineering, you have a capacitor resistor circuit, so CR circuit, LCR circuit, LR circuit. If you think these are certain modules which you can fit into the electrical system, you will get a sort of determined output, right? No matter how you design the system, if you put this module, you should get this particular output. Similarly, biology also has these network motifs and modules. And how do you operate? If you think in terms of a matrix, you can define something like this. You don't need to memorize this. I will just show you what this. For example, if you take a look at this put system, it's X activates Y activates X. Right? So if X activates Y and Y activates X, and this line is important. Let's assume all self interactions are inevitable. So X usually will degrade itself, or rather inhibit itself. It is not always the case in biology that a molecule A will always degrade itself. It can also enhance its own production. But at least for this matrix system, assume that interactions are inevitable. So if X is activating Y and Y is activating X, if I write a 2 cross 2 matrix, how should it be? So first one will be XX interaction, which you can assume as negative. It's an assumption. And say. Second one is how X affects Y. Okay? It's a plus. How Y is affected. If you take a this system where X inhibits Y, so the self comes negative, right? How Y is affecting X, it is not affecting. Right? But how X is affecting Y, inhibiting. So it's minus. So if I say this is XX interaction, this is how y affects x is x y. See? So this is how in a matrix format you can represent the systems. Right? Make sense? Now let us discuss some motifs next. Okay? And this might get a little complicated as you move across each motif. So stop me if anywhere it gets too complex. Suppose this R which you see is a protein. Okay. And let me define some basic terminology, at least for those who didn't have biology. I'll be using these terms regularly. If you look at the genetic picture in the central dogma, DNA gives RNA from RNA, or rather from DNA, RNA is made, and from RNA, proteins are synthesized. This is called transcription. This is called translation. We'll be using this one. So, oh. Suppose you have a protein R, which is produced at a certain rate. And since it's a molecule, of course, it will have its own degradation. And suppose you consider that some signal is there, S, which helps in the production of this. If I want to write the differential equation for such a protein concentration, if you have drdt, it will have some basal production rate, its inherent production rate via transcription. Then there will be some K1S term because of the signal. The signal is helping the protein to be produced. And you have a minus K2R degradation. Right? Okay. Now see, if I take this K2R and take a look at this one. So you have drdt on the y-axis and you have R on the x-axis. So if I want to plot this K2R, it is this black line which you see, the solid line. This drdt is a linear function of K2R. Ignore the negative, we are just considering modules. Okay. And if I plot this term separately, which is K0 plus K1S, it has no R dependence, so it is this dash. Makes sense? Now, if I want to find out the steady state level, I have to set drdt equal to 0. And that will give you the steady state concentration of protein as K0 plus K1S by K2. It's simple. It's not. So, how does this protein level, which is called as the response here, or you can think in terms of the steady state level of protein, vary with S, its linear level? K0 plus, so it's K0 by K2 plus K1 by K2 into S, y equal to mx plus. If you want to think in terms of this drdt picture, what is it? At the point where K2R and K0 plus K1 is intersect, that is the steady state, right? So the K2R is fixed here, and as you slowly increase this K0 S, or rather, if you slowly increase S, this K0 plus K1S will shift. For example, at S equal to 1, you will have the lowest dotted. When you put S equal to 2, you will have someone here. When you put S equal to 3, so as you increase S, your line of K0 plus K1S is increasing, and the point where it is cutting is moving towards higher values of R. So from both this point and from this graph as well, you see that as you increase the signal strength, the steady state value of your protein response also increases, and it increases in a linear fashion. So that's why this is a linear signal. Next. No, I plotted only the magnitude. So I plotted only the K2R. That's why if you see, if K0 plus K1 is, is equal to K2R, then this becomes equal. We can also have something called a hyperbolic signal response. Suppose there is a protein R and it gets phosphorylated. So I call it R. And what is in the first? So if you have idea of amino acids, there are few amino acids called serine, threonine, which have a free OH group. So the serine and threonine proteins, they react with a phosphate group and they become like this phosphorylate. So this process is happening. Definitely the source of phosphorus in biological systems is ATP. So ATP goes to ADP plus inorganic phosphate, and that inorganic phosphate will be attached to your protein. And suppose this process is mediated by some signal which you put in, which is this. And also this protein RP in the phosphorylated form can also undergo dephosphorylation, where the phosphate group gets reached. Okay. So if you want to write a system like this, where you have the concentration of the phosphorylated form, PRPT. So what is the production rate? It is definitely K1 times your signal strength times your free protein. And I can write the free protein as protein times phosphorylation. So this is your production rate of the phosphorylated form. And more you have phosphorylated, more will be the dephosphorylation. So it's A2 times. Now, if you want to plot this, if I plot the K2RP portion, again you have this solid black line, which is linear because it's a function of R. And now your production term, if you take a look at this, will be these dotted lines, right? And as you increase S, the slope will change. So at 2, you have this particular line. So when S equal to 2, when you put S equal to 4, the slope will increase. When you put S equal to 8, it will further increase. Right? So again, if you increase your signal strength, your steady state value of that phosphorylated protein is also going up. But if you take a look at this graph carefully, you will realize that this is not linear anymore. For example, if I have a graph something like this, where the line was something like this. So for let us take for S equal to 0.2. When I take S equal to from 0.2 to just 0.4, you see there is a large change in the steady state concentration of R. But as you keep increasing R, you see the gap between these points is gradually decreasing. So when your value of S was low, as you increase S from low values, your change in the steady state concentrations was very high. But as you keep increasing it, the saturation is achieved because even in changing S by 2 times, in the x-axis, your steady state level of R protein is not increasing proportionally. So I think from this picture, it is definitely clear that this is a hyperbolic. When you increase S in a low regime, you will get sharp changes in R steady state. But once you reach after a certain value, even if you change the signal strength by a lot, you won't get that much increase in your protein. Makes sense? And also, if you put this equal to 0, you will get the phosphorylated steady state protein at this value. So this is again Michaelis Menten if you can identify. So RT into S by some, which is a hyperbolic function. Now, even if you have the same network which is operative, if you model your equation in a different way, you can get a different signal response or response processing. Imagine, so the initial portion stays the same. So R is a protein R, it's phosphorylated in presence of some signal. So this term remains in there. But here, see earlier what I had written, I've taken a K1SS into RT minus RT. But here I have taken it in form of a Michaelis-Menten kinetics. And why I have taken it as a Michaelis-Menten kinetics here? Earlier I had assumed that this phosphorylation which is happening This is terms of a normal signal X. But usually, if you think of it, in terms of biology, usually this phosphorylation, dephosphorylation is carried out by enzymes, phosphatase, kinase, dephosphorylase, right? So since it is enzyme kinetic sort of thing, even if I keep this motif and just instead of normal A, if I think in terms of this process being enzyme mediated, so I can use Michaelis Menten kinetics. Right? So I will have a production rate of this phosphorylated form and also dephosphorylation term. Now, if you plot this K2RP and K2 plus RP, you get this solid black line, hyperbole. If you plot this portion, so this is also hyperbole, but there is a negative sign. It's a minus RP. So it is these dotted lines. Make sense? Okay. Now, if you take a look at how this cutting behavior is varying, initially you see when S is supposed to 0.25 and you increase it to 0.5, there is not much change in the steady state value of R. When you go from 0.5 to 1, there is a lot of change in. You go from 1 to 1.5, again there is a lot of change in series. But after that point, there is none. So there is no change, sharp change, again. So this is a sigmoidal behavior. When S is low, in the low regime, if you increase S, you see the RP values are not changing much. In the intermediate region, it is changing by a lot. And again, in the high region, it is excessive. So it's classic sigmoidal behavior, right? And see, this is a graded response, right? There's a gradation in the response. It's continuous and it's increasing. And definitely, this is reversible, right? If you increase S in this direction, it will go up. If you start from S value this and decrease S, it will come down. So it's reversible. How do we figure out the steady state concentration of this phosphorylated form of R? So you have to set this equation to 0. After rearranging, you get this. So if you set this to 0, you see, you take this to the right hand side and then you cross multiply. So you will get K1S RT minus RT into K2L plus RT and equal to K2 RT into KM1 plus RT minus RT. Makes sense? So if you solve this equation, you will get RT series. This can be solved by hand as well, and it was solved by two scientists called Goldbetter and Kostler. And the ultimate output, which comes as this output in steady state, is in terms of a function which is called the Goldbetter-Kostler function. We need not know the exact details of how the algebra or how the functional equation looks like, but since from at least from the graphical picture, we had managed to see that it is a sigmoidal picture. Definitely, this function is a sigmoidal function. If you want to see how to derive this by solving this equation, you can ask me. I'll share you the supplementary portion of this paper. This is a famous paper by Goldberg and Kistler, and they solve this problem. But for our purpose, let us assume that once you solve it, you should get a sigmoidal behavior of R with respect to S. And this, the Goldberg-Kostler function, generates something called a zero order ultra sensitive. Why is it called ultra sensitive? See, there was no change. Suddenly, for a very narrow window of your signal, there is a sharp change. So this is a ultra sensitive region. Again, for very small change of signal, you get a very high sensitivity of response in your system. It's called a ultra sensitive. Make sense? Okay. So this was a linear network motif, right? See, your signal, whatever is here, I've kept it as the linear. So this was a linear function of it. This was hyperbolic signal response curve, but still S was a U. See, this is sigmoidal signal response curve, but Still, I have kept this S. So input is still linear, at least in the S term, but its manifestation is coming as a non-linear. So it has become sigmoid. Earlier it was hyperbolic. With the same linear one, I managed to generate hyperbolic. I managed to generate sigmoidal one. Right? Okay. So you see that even if I keep the same motive, depending on how I model my equation, I can generate different outputs. Right? This is very important because you see, when you do a biological experiment, what is the outcome you get? You get this curve. You take some cells, you put some signal to them, and what you measure in terms of say fluorescence intensity of certain, if you have tagged some proteins, or if you want to quantify through some say blotting experiment or whatever, you do a Western blot to find out protein concentration, or you want to quantify it. Whatever you get as the experimental output in biology is the signal which you have given externally and versus this. So what you get experimentally is this picture. According to the picture you get, you decide how you want to model your equation of interaction. Suppose you know that protein X is interacting with Y. You do some sort of an experiment where you put in some external perturbing signals. You get a sigmoidal curve. Then you know that X and Y must be interacting in this fashion. If you get a hyperbolic curve, you know that X and Y are interacting in this fashion. If it's linear, in this fashion. So these curves are your experimental curves. And these will give you the idea that how should I write down my equation so that I can correlate it with my experimental outcome which I want. So this is how you model systems in systems. So let us take a look at some modes. This is something called a perfectly adaptive linear signal response curve, and I will explain. So, for example, you have some signal which helps in production of a protein R, and that same signal helps in the production of another protein or whatever bind. Let's assume protein as X. And this protein X degrades R. Okay? Or not directly, it helps in the degradation. So it positively affects the degradation of R. And definitely X will also have its own degradation. So if I want to write the steady state, or rather the equations for this system, if I want to write dr dt, it will be a K1S. Right? So it's being synthesized at this rate. And there is a minus K2XR. So more the R, more it will be degraded. More the amount of X, faster it will be. For simplicity, you can assume that you can keep a K naught here, it won't change the behavior. And we can write dxdt as it is being produced by this K3S, and it has its own self degradation. So if you put both equal to 0 and solve for steady state, you will get the R steady state value of protein R as this, and for protein X, you will get you can try. It's pretty easy, right? It's just two equations and just two. You said both equal to 0, and you will get these two steps. Now, if I want to do this DRT versus R curve, which we had before, take a look here. See, this negative term is K2 into X into R. So it's a linear function of R. Right? So it's straight like this. However, it is not only dependent on R, right? It also has a X dependence. And X is directly dependent on S, right? So if you think carefully, this K2XR term, if I increase S, the slope of this K2XR term should also go up. Make sense? See, R is definitely increasing, so it's a linear function R, but the slope is K2x. And K2x is proportional to x. So the slope is proportional to your signal strength, right? So from if you have s equal to 1, the curve will be something like this. If you have s equal to 2, it will be this. If you have s equal to 3, it will be this. Okay? And again, we are plotting the magnitude value. So don't worry about the negative value. Alright? And if I plot this K1x curve, it has no R dependence, so it will be a flat line. But it will be a stepwise line. So when you have s equal to 1, it will be something here. When you put double the signal strength, it will be something here. When you put higher signal strength, it will be something here. Okay? Right? Now see, no matter what your signal strength is, your steady state value of R is constant. Right? And from this also, you see it's K1, K4 by K2. It has no dependence on your signal strength. So no matter what signal strength you put in, the steady state value of your response, or here the RQT, or some you can think this in terms of a response as well. The steady state value of that is independent of the signal strength you put. That's why it's called a perfectly adapted linear signal response. Whatever signal you put in, it will adapt to that and ultimately reach the steady state no matter whatever signal strength you put. So it's an adaptive system, right? If you want to look in terms of a dynamic picture in the temporal dynamics, if you plot the concentration, or rather the X concentration, R concentration, or the signal strength with respect to time, see, if you put signal in terms of pulses, once you put in S, you will see an initial spike in R because S is producing, right? However, during that time, X is also increasing. Follow the green. Okay? So initially R will increase, but once X increases, it will start bringing down R. Okay? So it will come down again. Again, you put a pulse of signal. Again, R will grow, but once X goes up, it will bring it down. Again, you put a pulse. Again, you put a pulse. Again. So you see, no matter how much signal strength you're putting, your steady state value is remaining constant. And this is called a sniffer response method. The name should suggest something. What do you think it suggests? It's in the name, right? Sniffer. So this is how we sniff things. If you go to the chemistry lab, if you do an experiment which produces H2S, as soon as H2S is produced, you will get a strong sensation of the smell. If you allow enough time to adjust yourself to that smell, it will decay down and you won't get that signal. Again, if you have a burst of H2S gas, again you will smell it. But if you allow yourself to adapt it to a certain time, it will again go down. So it's an adaptive behavior. This is how we sniff things. So this is called a sniffer response behavior. That once you have the signal, there will be an immediate effect, but that effect will die down and the system will adjust itself. Make sense? Now let's take a look at something, and this will be one of the most important things in the context of systems biology, which is called the positive feedback. The little careful. For example, you have a signal S, which is synthesizing R, and R has its own degradation rate. What R does is R itself helps in the phosphorylation of a certain enzyme or some species enzyme you can consider it. And that phosphorylated form also helps in the synthesis of R. Possible, right? So essentially, the basic motive of this is something like this, right? R enhances the production of EP. EP enhances the production. So it's a double positive feedback. So if I want to write the differential equation of this drdt, what will it do? It will have some k naught into EP into R. EP, not into R, rather EP R. And what is this EPR? We are using the Goldbetter question equation, which we had mentioned. So if you take a look at this, remember when you had some signal producing this, I had written in terms of the signal responses. So here, instead of a signal, so EP is an internal signal to the system. So it's a EP and this particular article. So it's a Goldbetter question. Okay? And then you have a synthesis rate, which is K1S, and you also have a degradation rate, which is K2X. Assume that there is something X which is helping in the differential. Alright. Makes sense? Now, again, we do the same sort of plot. If I want to plot drdt as a function of R, if I plot this K2XR term, it will be this linear solid black line. If I take this entire term, which is K1S plus your Goldbetter function, the Goldbetter function is sigmoidal. Okay? So, if you have S equal to 0, it is something like this. Right? If you increase S, the entire function will be just shifted, right? So it will still be sigmoidal. It will be somewhere here. So you see one going from 0 to 8, the entire function is just shifted. So initially it was k naught EPR, now it is 8 k1 plus k naught EPR. Then it is 16 k naught plus k naught EPR. Right? So it's just shifting the sigmoidal part or this k naught EPR plus this positive part upwards. And wherever these two lines intersect, that is your steady state, right? And the corresponding x point will be. So now, do you understand how this picture comes from this? Not very evident, right? Okay. I have taken a representative sigmoidal function, which is this 1 by 1 plus e to the power minus something, e plus s. This signal. And I have taken that k2 is 0.125. Now, if I gradually increase this, okay, can you see two gray dots? At least three gray. Ignore the gray dot which is there in the x equal to 0, so the y-axis. Apart from that, you can see three dots, right, where it is coming. So there are 3 points of intersection, which means there are 3 steady states. If I increase this further, you see two of the steady states are coming together and one vanishes. Also, oh, sorry, two steady states come together, the lower one and the upper one steady. When I'm at this point, what is that? If you remember, since two steady states were there, and now it has merged into one point, this is a half stable point, right? And if I increase this further, the points don't exist anymore. So this point here, what sort of a bifurcation point is this? See, if I draw it again, if I keep it here, initially there is no point. Suddenly there is a half stable point. Beyond that, there are 2 points. This is a saddle load bifurcation, right? I had shown, remember, saddle load initially it is out of the blue, blue in the sky bifurcation. There is no point. Suddenly you see a half stable point. Beyond that, there are 2 points. So, to think carefully, let me keep s equal to 0. Okay? You see, initially there are 3 points. So this lower steady state and the middle steady state, they actually come closer as you increase s, right? And at this point, it's a saddle. Beyond that, these 2 points don't exist. But you see, the higher steady state, which is this extreme right hand graph, that is always present for positive values of s. No matter where I put s, here the first and second states don't exist, but the third exists. The third still exists to appear. The third always exists for some positive value of s. So the third should always exist for all values of s, and the first and second should collapse at some point, some value of s. Lesser than that, they will exist. More than that value of s, they won't exist. And as I increase s, the distance between those two steady states should decrease. And also, if you look at something, for no positive value of s, am I able to get rid of the third state? Now take a look at this. For all values of positive signal strength, the upper steady state exists. As you increase your signal strength, the distance between these two steady states each decreases. And also I must mention that this is stable, this is unstable, and this is stable. If you want to do it, it can be done, but we have to just put this equation to 0, and once you do the Japon stability matrix Dilip, you find that. So this is, that's why this is a stable, this will be unstable, this will be stable. You can try it yourself. Or if not, I will refer you to the okay. But what is important is, as it increases, the distance between these stable and unstable states, the first and second steady state is decreasing. At some critical S value, there was a saddle node. Beyond that S value, the states did not exist. Right? And the upper steady state always existed for every value of signal strength. Right? So now does this picture make sense from this? Right? So if you think carefully, suppose you have started your system somewhere here. You slowly increase the signal strength you are putting in. After this point, your system will jump here. Now, if you decrease your signal, the system never comes. Yeah. Suppose you start your system somewhere here at very low signal strength. You gradually increase it. It's in the low, low, low. As soon as you cross this point, it will jump to the higher steady state. Now, from that higher steady state, if you start decreasing your signal, it will remain on this, and your signal, even if you take it down to 0, it will not. So, this is a irreversible switch, because your system switched from one state to another state, but If you take the reverse path, it did not switch back. That's why it's irreversible switch. Okay. Makes sense? If there is a irreversible switch, there also has to be a reversible switch. Right? So take a motif like this. It's the same process. So S helps in synthesis of R helps in phosphorylation of a protein or enzyme. And now, instead of the phosphorylated form helping in the synthesis of R, you have the non-phosphorylated form or the unphosphorylated form actually helping in the degradation. So essentially, R provides a negative feedback to E, and E also provides a negative feedback to R. So it's a double negative feedback. And how can we write this equation? Again, if you want to write dr dt. So now, as you were mentioning earlier, you can take a Bessel synthesis. In the earlier case also, you can take a Bessel synthesis. It will just keep shifting your curve. So K naught plus some K1s term, then it has a inherent degradation rate, which is K2R. And also, there is a E-mediated degradation. And again, we are using that E-mediated degradation in terms of a gold beta quotient. Okay. So now, if I plot this entire thing, this K2R minus K, or rather, if we take that, so minus of K2R plus K2 prime ER into R, you will get a curve which is like this. It's a, you can try plotting this on any plotting software, you will get a curve. And if we have the K0 plus K1s, again it will be a flat line, and depending on S, you will just keep shifting this line upwards. So this is 0.6s, there's something like this. If you increase S, it's 2. If further increase S, it is right. Now, see how is the behavior. When you have no value of a signal strength, you are at your lower steady state. When you increase to some intermediate value, you have 3 steady states. This, and this. The first and the third are stable, and the second is unstable. If you further increase your signal strength, the first and second one vanish, and you have only the third one. So definitely, the picture should look something like this, right? When you have low values of your signal strength, only the lower steady state is there. Once you slowly increase, you go from here to here to here to here. You see that the distance between the first and second is actually decreasing. So it's higher, then it decreases, and between second and third is increasing. So it's small, it's large, it's large. And once you cross this particular value and you go beyond that, only the higher steady state. So see, beyond this is critical field, only at the highest. So there are two critical points. And what are these points? These are 7. Right? Because you see, if you look at this portion, initially there was no states. If you just as soon as you reach here, it will be a half stable point, and beyond that there are 2. So this is 1. And if you go from top to bottom, initially there was no state. There is a half stable point here, and then 2 states. So both of these are 7. You see, at some intermediate region, you have possibility of both the states exist. Right? In this particular instance, both the states. So the system can be in either the stable states. Both the states are in stable. Now think of this. You start off your system here. You increase, 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 increase. It's still in low. Once you cross this, it jumps over here. Now you decrease, 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 decrease. It stays in the higher steady state for a much longer range of this. Only then it comes. So essentially, the amount of signal you need to put in to switch the system from the lower to higher steady state is larger. The amount of signal you need to put down, or rather signal the change you need to take it from higher to lower state, is asymmetrical. It won't switch down at the same point as it switched up. This is definitely a hystericity. The two parts are not exactly. This is called the topology. And we'll discuss a very good example of this and how this has been proposed by some modelers, mathematical sort of biologists, but no experimentalists believed them. Then after 10 to 12 years, it was actually shown that this sort of behavior exists. And I'll show you how that behavior looks like. Okay. Now we had been talking of some abstract terms that E, R, E, G. Let's take a real life example. So there's a certain transcription factor. The transcription factor is something which helps in the transcription. So once it binds to a transcription factor, PPF is one such transcription factor. And transcription factors of protein, they can be obtained easily. So one such protein is called E2F. It is a transcription factor. Okay. And how does it do? So you have the mRNA of E2F. From that, protein of E2F will be mixed. And this E2F is special in the sense that this E2F protein can enhance the production of its own mRNA. So, if you look, essentially it is something like this, right? From mRNA, protein is being produced and protein is also energy. If we consider the protein concentration of E2F, okay, so you can write some DAGTP. Without getting too much into the details, you can write some sort of equation. Like so you have some basal translation rate. You also have some basal degradation rate. And this backtrack positive feedback is in sort of this particular. Okay. Now, why is there x to the power y to the power n? We could have just written something like not into protein concentration. Why into x to the power? If you think in terms of biological perspective, remember I had given the example of hemoglobin binding to oxygen, right? There also we use a Hill kinetics, right, of hemoglobin binding to oxygen. We have partial pressure of O2 to the power n by some Kn plus Why do we include the N term in case of hemoglobin? Because hemoglobin is a tetramer. So whenever you have polymer formation or oligomer formation, usually the system kinetics follows some exponential determinant. So here, this protein is activating its own sort of mRNA or pieces of its own mRNA. The monomeric form of EQF does not exist. Once EQF forms a dimer or a trimer or a tetramer, those forms actually carry out this process. That's why we have taken a to the power 8. Now, if you write the equation is now, what is the biological significance of this equation? If you remember your cell cycle for those who study biology, the cell cycle has certain distinct phases. Right? There is a resting phase, which is called G0. Then the cell is absolutely dormant. If it enters the G1 state, it is the first preparatory growth phase where it builds up, it increases in size, it starts to build up and gets ready for division process. After G1, we have the S phase. In the S phase, which is the synthesis phase, here DNA starts to get duplicated. Then you have the G2 phase from S to G2, where again some further growth and processing and preparation. Finally, in the mitotic stage, the cell divides and the new daughter cell again goes to the same cycle of G1, S, G2. What this EQS protein does is it acts as a checkpoint protein in this G1 to S checkpoint. So, when the value of this EQS protein is low, the cell will remain stuck in the G. Only when this EQS protein is sufficiently high, the cell will switch from the G1 to the S. So, what it looks like in biology is something like this. Okay? If you don't have, or for example, if you have a signal strength, but you don't have enough value of N, take my word for it, it happens here. If you don't have N, or you don't have sufficient non-linearity in the system, so why am I calling it non-linearity? Because if you have N equal to 1, it becomes a linear system, right? And for very low values of x, you can say that N plus x, we ignore the x, so x by k. So it is 0 plus Gx minus 2Gx. So it's all linear terms. But once we have h to the power n, it's a non-linear system. Make sense? So if your value of n is not very high, say for example n equal to 1. If n equal to 1, your steady state concentration of x will definitely be a linear function of the signal strength. And in terms of signal strength, so you can either call this V0 as signal strength or mu as this. But if n equal to 1 and if you ignore this kn plus x, assume that, then definitely you can see after setting this equal to 0, x will be some linear function. As you slowly increase the value of n, you're starting to develop more and more non-linearity in your system. For example, if you have x equal to 2, once you start to solve, you have to solve a quadratic equation. If you have n equal to 3, it will be a cubic equation. So definitely this x versus x steady state versus signal curve won't remain linear. What you'll see, you'll start to get this sort of intermediate kink in your system. And once you have sufficient non-linearity in your system, okay, before that, once you have sufficient non-linearity in your system, the graph will change. If you take this stage where you don't have sufficient non-linearity, for example, if you change the signal strength, you will see a graded change, right? It will be a sort of uniform change. It will be a gradual change. That was the graded response, basically. If you have sufficient non-linearity, what happens is the system bifurcates into a toggle stage or a bistator stage, which I showed. And this lower state is definitely the low EQF state. The low steady state is the low value of EQF. And that what has been called is the EQF off. If you, because it's a switch, right? So we have switch off and switch on. So the low state we are calling a switch off. And when the EQF is in the high state, we have called it as EQF on. So the EQF will switch from off to on state. When it is off, that means it is low. So the cell is still stuck in the G1 phase. Once you switch to the high value of EQF, the EQF is switched on, you transit to the state SDPF. So the low EQF determines the G1 phase, and the high EQF determines the SDPF. Now, earlier you had a graded responses. As you increase this, there was a uniform change. It increased almost monotonically. How will the signal change, or rather the steady state of this particular protein change if there any variations? We start from a low signal, it will go to, once it reaches here, it will switch to the higher level. Okay. So, and when it's in the higher level, it will come back here, it will come back to a much lower point. Then it will transit to the lower level. So, and the intermediate region is a bistated. Okay. This was proposed in terms of mathematical model, but no one believed that this can be possible. That if I increase my signal strength, how can I have such a hysteresis in terms of a protein? If I put in more signal for synthesis of my protein will go up. If I decrease the signal, protein level will come down. Why this sort of, where is the bistability? Where is the hysteresis? How is it possible? No one knew. But it was actually shown in experiment. It was done by Yao et al. It came out in Deciphering biology. It was something like this. So I want to explain to you in terms of, we discuss this experiment in detail once again when we do proper cell cycle modeling and stuff like that. But think it. If you have a marker for ETF, then, and it's a fluorescent marker, then you can measure, if you take suppose the 10,000 cells and you put a fluorescent marker for ETF, then you can measure the histogram of your fluorescent synthesis, right? And how many cells have low EQF, how many cells are in 10, how many cells are high. And how the experiment was done is, see, initially the cells are in G0 dominant state and neutral state. You put a pulse of serum, so you put a sudden high dose of serum, and that will transit cells to the G1 state. And now, if your pulse is strong enough, your EQF will also be high. If you put a weak pulse, your EQF will be. So, your signal strength is your serum pulse condition. Now, if I explain this experiment to you, what they observed is when they have low signal strength, they found only, I think this is EQF increase in this direction, and this is frequency. That's how histogram looks. Initially, it was very low signal. When they put intermediate serum pulse, what they observed is there is a bimodal population. So there are two distinct classes. Some of the cells are low EQF, some of the cells are high EQF. When they put an intermediate. When they put a high pulse, you observe there are only high EQF. Makes sense? This experiment, in the direction I have made, what they do, it is low to high. For example, if I say this was at 0.5% serum, this was at 2% serum, this was at 10%. When they did the reverse experiment, at 10% serum, It was something like this. When they brought it down from the high to the 2% serum pulse, what they observed is more number of cells were still in the high phase compared to the low phase. If you look at the increasing serum picture, you see I had drawn that at 2% there were more number of low cells and less number of high cells. In the reverse direction, even at 2%, what they observe is there is no population of low APF cells and high population high cells. And when they brought it down to 0.5%, well, so if I take, suppose this as, let me say, for example, there was still a population. Only when they dropped it back much further did they get perfect blend. This is only possible when you have this. Right? So if you go from low to high, at very low you will have only low APF. Then at intermediate you will have low and high. And when at high, you will have only high APF population. If you do the reverse experiment, at high you will get high APF population. When you bring it back, you will get high APF, low APF population. But you have to bring it down to a much lower value of serum pulse. Only after that will you start seeing perfect low. Next slide from this picture. This is never possible if you have. If you have this, what do you expect? When you put low signals, you will see unimodal. And once you slowly increase, you will not see two distinct populations anymore because this was a switch, right? So it's a 0-1 sort of situation. This is a monotonic increasing point. So the peak will keep shifting in a gradual way. You won't see low and high itself. It will be low, slightly more, slightly more, slightly more. It will be a monotonic shift in the bistable case, you will see one population going down, one population coming. And this will occur in a different difference. Because in the low to high direction, the amount of signal you need to put to get significant amount of high population. In the reverse direction, you need to go back to a much lower jump to start getting the only more amount of low. So this was one example of how a toggle switch or a bistable switch is operating in a real environment. One more example, I'll discuss this is very brief. Instead of double positive, you can have a double negative resistance, which I showed earlier that this R was like phosphorylating, so it was decreasing, and you are also decreasing. You can write it in terms of somewhat like this. Okay, yes. So in this case also, what you will get is a toggle switch. Because think carefully. X is inhibiting Y is inhibiting X. Right? So in a sense, isn't X activating itself? You are decreasing something which affects you negatively. You are decreasing something which harms you. So in a way, you are increasing yourself. Right? And in a positive term, you are increasing something which benefits you. So essentially, you are also benefiting this. So a double negative is also positive. Makes sense. So positive into positive is negative into negative number. So in this case also, in case of a double negative as well, you will get this sort of toggle switch. Now, remember I had shown you that irreversible switch means irreversible switch, if you think carefully, in a real biological sense, if the switch exists somewhat like this, but one of the saddle nodes mathematically is in the negative signal range. Okay, then these are one-way switch, right? Because you cannot put negative signals in binary. So it is not always like the motive is perfectly reversible or perfectly irreversible. It depends on the context. Because remember, what I said is that this you will get experimentally. Right? If you get this experimentally, then you go back and know that what I should do. That's my model equations. But always be careful that whether it's an irreversible switch or it is actually a mathematically reversible switch, but one of the domains is in a non-achievable domain, which you cannot practically achieve, right? So a reversible and irreversible switch might differ by the position, and that will obviously depend upon parameters. I can show you one example. Remember this particular example which I showed? So 0.15. So if you remember, this was what I had shown the irreversible switch, right? So, so initially you have only the high level of steady state. See, if I keep decreasing S, there are three steady states. And if I put S equal to 0, right? 0, then there is the, if I put S equal to negative, you see that the lower steady state also exists. But because it is at negative signal strength, it was not achievable practically. But it exists mathematically. Right? If instead of 0.125, I put it as 0.15x, you see there is a high steady state. If I decreases, there are three steady states. So if further decreases at some positive value of it, you have the only lower steady state. And this is a reversible situation, right? Lower steady state, saddle law, unstable, saddle law, high steady. If I just, so what I did is nothing, I just shifted the value of this parameter and that converted the switch from a reversible to a irreversible. So everything depends upon how you model your system and what parameters you use. Okay. We'll take a short break here for I think 10 minutes, and after that we'll discuss something which are how oscillation system is Okay, so we are back from our short break and let us discuss something. So today what we have discussed, which is a linear response, hyperbolic response, then further we moved on to this positive feedback, negative feedback, and bias fidelity. If you think in terms of the long-term behavior, what we are saying is that these all ultimately reach a finite value steady state. After at a steady state, it will reach a finite value. But not all biological systems operate in such fashion. So, for example, once I show this to you, it will either be low or high. But if you think in terms of like circadian clock, for example, your body follows a nearly 24-hour cycle. Right? So there is certainly must be some genes, some proteins, which also have to follow a 24-hour clock. So it has to go up, come down, and that's how your body will keep adjusting that, okay, if this is day, I have to follow this, if this is night, I have to follow this. Definitely, there are certain oscillators in biology. If we follow some example, for example, I mentioned the circadian rhythm. It is controlled by certain genes called per clock. Names are not important, but they have to oscillate, right? Because that's how a clock operates. You have to oscillate with a certain time period. There are certain signaling, such as calcium signaling. You have metabolic systems which oscillate. And you see there is a wide variety of the time periods with which they oscillate. For example, if we had mentioned cell cycle, right? So G0 to S G0 to G1 to S to G2 to S. Now, if you want to cross over to certain states and then restart the cycle for your daughter cell, definitely there has to be some sort of oscillation. Right? Something has to go up and come down, go up and come down, and that's how you will enter a state, exit that state, enter the next state, exit the state. So this is how. So there are certain oscillators in biology. Now, how do oscillators function? Suppose we have a system like this, where a protein is being synthesized. It also has a certain degradation rate. And that protein also inhibits its synthesis by a cell. Okay. So if I denote this protein as Y, I can write dy dt as some, you put some signal here, this fearless. Then you put a hill term. Assume that after the protein becomes a dimer or trimer or tetramer, only then it inhibits its own formation. And the degradation term, you assume that there is some enzyme in here degrades the thing. So you use a Michaelis Menten for that. Now, if you do the same sort of pH, which is you put the K2ET Y by K plus Y, that is the red line, this straight line. And if you plot this, it is the green. Right? Try plotting this. Okay. So, wherever these 2 points intersect, will be a steady state. So, this is a steady state. Okay. Now, I will leave something, or I can show you, because here we have not taken any sort of values for QoS, right? So it's difficult to understand. If I start somewhere, how will my system be? You can take some examples to generate a graph like this, put in your own parameter values, and if you remember from the first class, or rather the second class where I had shown that how do you calculate the dy dt and dx. So you just calculate the dy dt and start drawing arrows here. Okay. So what you'll see is, and if you remember how I had shown in case of the predator framework, that these lines are the zero lines, right? So where you put the dy equal to 0 or dx equal to 0. This is one of such lines. So as soon as you cross over from here, either the x dot or the y dot value. Since this is a one-dimensional system, you can consider only y dot. And you see in some of the regions, this will be positive, it will be negative, collapse. You can put some values and check, you will get this sort of a behavior. So this, if you see, is definitely a homeostatic behavior. No matter where you start, what is homeostasis basically in the system? That no matter whatever perturbation you put in your system, ultimately the overall balance, which is the ionic balance, the osmotic balance in your body, is maintained at a constant rate. So that is the homeostatic state of your body. No matter where you start, everything comes back to this. So this is definitely a stable load, right? Now you do something, that is, consider that there is a time delay in the system. What do I mean by time? Here I had mentioned that this protein is formed, and in its dimer or trimer form, it provides a negative feedback to its own formation. Since it is inhibiting itself, it's a negative feedback. Right? It's providing. Assume that there is a certain delay, which is also practically feasible, right? Because once a protein is formed, it needs some time to form a dimer, a trimer, a tetramer, right? Only after that time delay will it be able to provide the negative feedback tubes. So if we assume that there is this tau amount of time, which is suppose say the formation time, the amount of time you have to allow for the sort of polymeric form to emerge, then your equations will be modified in this particular. You see, the degradation term is still in the t times. So wherever t is 0, degradation is operated because as soon as a protein is formed, there is a chance that it will get degraded. So that is in the t time scale. But your negative feedback is now in the t minus tau time scale because you have to allow t time only after that. So the difference between your tau and the time elapsed, only that much time has been made available for the polymers to degrade your synthesis or degrade the protein or stop the synthesis. So now you see what will happen is that this particular rate and this particular y, so this is a function of t, right? This is also a function of t, but here you have put an additional tau dependence. It might not be exactly visible in this particular graph, but if you solve this using some solver, you either use MATLAB or Python or whatever you want, what you will see is that you will actually start to see oscillations in your system. Why is it so? Because in terms of this particular vector field picture, instead of this being a stable spiral, your system, what it will do is, since there is a time delay, the exact response of y is not instantaneously reaching your system, unlike earlier. So because you have separated one factor by a tau time, your system will actually overshoot in one half, this half, and undershoot your stable load. So it will not spiral like this. So essentially, to spiral like this, you need your degradation and inhibition to happen at the same time. But when you have separated one process by some tau time, the system will go around this. Think physically. If you have a time delay, if you don't have a time delay, whatever is being produced, it is instantly being inhibited. So it is like a steady state situation, right? But if you have a time delay, you allow certain amount of protein to fall. Only after that you start degrading. But once you have degraded it enough, you don't have any sort of protein monomers, protein polymers or the aggregate forms left. So again, some protein will be formed. There will be a, because you have the delay operated. Only then after some protein has been formed, again the aggregation will happen and it will bring down. So this time delay allows for sufficient time to increase your protein and then bring it down rather than inhibiting it from the first step itself. Makes sense? It's similar. For example, when you apply the steady state approximation, your intermediate concentration spikes up and then reaches a steady state value, right? But if your second reaction starts certain time after the first reaction, your X will go up. Only after certain time it will come down, right? But when it has already come down, the second reaction doesn't occur anymore. You again have to allow that tau time for the intermediate to build up again. Then it will come. So this is how with the time delay oscillations are done. Make sense? Now to do something which is you take a time delay system, so it overshoots and undershoots systems. Okay? What you do is you take a dimensionless value of your time delay and a dimensionless value of your C. And how do you define it? Okay, if you take one quantity which is K1S by K2, for example, see, rate constant, units cancel out, substrate concentration or signal concentration in the concentration of the concentration. Now you can rearrange this in terms of Kd by K2 and Kd by K1S. Just some mathematical joke. Now think carefully. What is your Kd by K2? Kd is something which is common in both the cases. Okay? So you put it in, but just for simplicity, so Kd you can cancel it, but that will give you the original equation. Right? But you have 1 by K2 and 1 by K1S. Right? Think carefully. If K2 is your rate constant or somehow involved in the rate of the degradation of the protein, 1 by K2 is your time scale of protein degradation. For example, how you used to do in radioactivity, 1 by K is the average lifetime of the radioactivity. So this K1 is by K2, and in terms of 1 by K1 is the synthesis rate of the protein. 1 by that will be the time scale of your protein synthesis. So this K1S by K2 is a dimensionless quantity, and in this particular context, it is also somehow a ratio of your degradation time scale with the synthesis time scale. So can I in essence say that this is a good measure of my signal strength? Since K1 is by K2, right? Higher the value of S, higher will be this particular quantity. And to non-dimensionalize it, to get rid of the units, we have converted into the time scale form. But inherently, we started with S, right? So it's still a measure of S. So higher the value of S, higher will be this. So this is a non-dimensional signal state. Use a similar thing. If you write E2 ET tau by Kd, so ET and Kd are both concentration units. K2 is rate constant, tau is time. So it's still non-dimensional. Right? And if you do the same generally, it's T by tau degradation. So this is a dimensionless quantity, which is a measure of your delay, and it's the ratio of your time delay by the protein degradation times. So now you have a measure of your signal strength and you have a measure of your time delay in unitless form. And what you can do is you plot this non-dimensional signal strength on your x-axis and this non-dimensional time scale on your y-axis. And then these colored lines which you see are nothing but the projections of your steady state onto this parameter sheet. I'll show you with one example. Suppose we say, for example, instead of a 2D, if you do a 3D, here you have A, and on the Z axis, you have the steady state. There are two parameters. Suppose the system is a shows a toggle switch type of bistable bifurcation with respect to A. So whenever I showed you that bistable diagram, I plotted with respect to one parameter, which is that means during that time I have kept my B fixed. So at that value of B, suppose the steady state is will look somewhat like this. At a different value of B, let's say when B is much closer to 0, the system will get adjusted in such a way. It might happen. You're still plotting with A, but now you have changed B. It might happen. If you plot for a larger value of B, might be that the picture is like this. Makes sense? And if you put B equal to 0, suppose it is not bistable, it just becomes like this. Which might happen, right? Because remember I had changed it from 0.175 to 0.125, and that changed from irreversible to reversible. So it can also happen that changing some different parameter, the entire bistability values. For example, if I change the n coefficient, if I put n equal to 1, it won't be bistable anymore. If I put 2, it will be bistable. If I put sufficient. So look carefully. If I plot the AB plane only, can I draw something like this, where these two lines are the locus of these two saddle lines? Okay. So whatever is inside this window is a bistable window. Whatever is outside is a monostable. So this is called a two parameter bifurcation diagram. So it allows you to check that how do two parameters control the regions where your system will show a particular sort of behavior. Okay. Now come back to this one. Your time delay and signal strength, essentially we have non-dimensionalized it, but they came from these particular parameters, right? It has K1, K2, Kd in total. So this time delay and signal are two parameters for your game. Okay? So this is also a two parameter bifurcation diagram. And if you put some numeric values to the system, for example, if you put P equal to 1 and Kd by Km as, so this is Kd and this is Km and different value, whatever is here, so away from the origin on the line, that is your oscillation. And whatever is below a particular line, that is your homeostasis, which I showed you. So for example, for 3.33 Kd by Km and P equal to 1, if your point is somewhere here, the system will oscillate. But if your point is somewhere, let's say here, it won't oscillate. Okay? Therefore, as you are changing the value of this Kd by Km, you are gradually shifting the regions where your system boundary exists between an oscillatory and a homeostatic. Okay? Now, when you have P equal to 1, let's say, and take any one example of Kd by K. Okay, let's say Kd by K equal to 5, the green line. Right? See, you need a lot of time delay beyond 5 and above to see oscillation in your system. And also, you need a signal strength of at least 1.5. Make sense? When you change the value of P1 to P2, you see every curve has now shifted downwards. So, for example, if at kd by k, I have taken 5, right? Okay, so 5 will be somewhere here. The graph should be. You see, now you need much lower time delay and almost the same signal strength. Therefore, from this, do you understand that to bring in oscillations in a system, you need two things. Either you need sufficient time delay or you need sufficient non-linearity. So this is one example. Suppose for this p equal to 2 and kd by k equal to 10. So you see that this tau by tau degradation, which is the signal strength. Sorry, this is the time delay. You need at least 2 and higher. And this signal strength, which is tau degradation by tau synthesis, you need at least 1 and higher. One example which I'm showing. So you need to cross beyond this point to get oscillatory. So the bottom line is this: to have oscillations in a system, either you have to put sufficient non-linearity or you need to put sufficient time. And how do you know that what is sufficient? You draw two parameter bisection. Now, see, instead of putting a time here, what you can do is there can be also a series of interventions, right? What I mentioned earlier is that suppose a protein is being formed, it needs sufficient time to aggregate, and only those after the time delay, after the aggregation, it can provide aggregation. It can also happen instead of a time delay, if you have a mRNA and a protein, that will also serve as some sort of a delay, right? Not in time, but in terms of the sequence of formation. Okay? So if you have an mRNA which forms protein, and you have that protein which inhibits the mRNA formation, then you can write equations and what here is if you just write this particular normal equation, you will again fall back to the homeostatic. Because here, although you have a sequence, they are you have to have some sort of separation, right? Earlier you had a time separation. Here you have a sequential separation. But still, you have considered that your system is in a confined volume. So, okay, the chance of events, even though they are sequential events, the chance of them occurring simultaneously is very high. If they are occurring in the same volume, as soon as one thing is produced, the chances, even though it's sequential, immediately it might happen. What you can do is instead of separating in time, you can separate in space. Okay? Spatial separation. Which can happen mRNA protein is nucleus again. This makes more sense practically in biology. Where does mRNA exist? Majorly in the nucleus. Right? But where is protein formed? By the ribosome. Right? And it exists on the endoplasmic reticulum in the cytoplasm. So once the protein is formed outside the nucleus, it has to somehow be trafficked inside the nucleus. Then it can start acting on this mRNA. And also, from this mRNA, the protein has to be formed. For that, the mRNA has to go to the ribosome from the protein. So definitely there is a compartment separation. in terms of nucleus and cytoplasm. So we have separated into a sequential process, and because we have put an additional compartmentalization in the system, now you definitely have a proper separation of it. When if the protein is being formed, it has to be first transported into some other place for the effect to do. So without inputting a time delay, in explicitly seconds or minutes or hours, we have put a sequential delay or a time delay, but in terms of the occurrence of events rather than the time scale. And if you draw two parameter bifurcations for these systems as well, you see these regions when you have sufficiently high values of P and this KD by K, that will separate your oscillation versus your homeostatic domain. Similarly, what you can do is you can also draw this parameter of half-life of your mRNA in the nucleus and half-life of your protein in the cytoplasm. Because these are also parameters. Instead of the rate constant or the dimensionless these particular constants, if you just take the half-life, if you assume first order 0.620 by 2, then you can, those are also parameters, right? If rate constant is a parameter, the inverse is also. If you do in terms of this, you will get a oscillating region somewhat like this. Now, think physically, what does it mean? If suppose you're in this system, what does it mean? Your half-life of mRNA in the nucleus is very small. Your half-life of your protein outside the nucleus is also very small. So, whatever mRNA is being produced in the nucleus, it is degrading so fast, it is so unstable, because its half-life is small, so that means it's unstable, that before being trafficked outside to have any significant effect, it has already degraded by. Similarly, in terms of your protein, if your protein is so unstable in the cytoplasm that it cannot be trafficked at all, then makes sense? In terms of physical picture display, if your these are so unstable that before being trafficked, they have already reduced by so much, then how do you put in the, how do you allow it to come up and the negative feedback to start becoming sufficient? Right? So only when you have sufficient stability of the mRNA and sufficient stability of the protein, will they be able to grow up to a certain extent, then be degraded, then reach. So essentially for oscillation, you need to have sufficient non-linearity, or you need to have sufficient time delay, or not rather, or I should mention and. So you need to have sufficient non-linearity and sufficient time delay, and from this picture, it's also clear that you need to have a proper balance between your time. If one process is too fast and one process is too slow, see, for example, this picture. Suppose your mRNA is very stable in the nucleus, but your protein in the cytoplasm is not stable. And how will it ever provide a negative feedback here? Similarly, it goes for you. If your mRNA is not at all stable in the nucleus, but your protein is very stable, then also there is no point of so sufficient non-linearity and sufficient time delay and proper relation between the time scale. Then again, we have one more example. This is a hysteresis dividend. So when you have a this particular, so this is what I showed you here. It was a stable rate sort of system, right? But this has the chance of falling back to this particular state if some region exists. So if you have improper balance, now biology has systems to control that as well, where you can broaden this range of oscillation. So what you do, you provide some hysteresis to it. Earlier, remember what the picture was. If I draw, earlier it was something like this, right? In that case, if you draw the vector field, it was spirally linear. Okay? But if you put some positive feedback into the system, and how will you put the positive feedback? See, this mRNA is produced with protein. Protein is inhibiting the mRNA. So this is the negative feedback. Okay? And this protein was being degraded by some enzyme. And who degrades the protein? The protease enzyme. If this protein is somehow able to inhibit the activity of this protease, then it is stabilizing itself. So it's a auto positive feedback. So if I draw something like this, if I have mRNA, mRNA activates the protein inhibits the enzyme. Right? Right? So now you have put in some positive pushback to the protein. And the line which was here earlier becomes something like this. And now, again, what will happen is instead of a stable spiral, if you draw the vector field, solve this immediately, you'll start to see stable oscillation. And this will be steady. So you will again start to overshoot, right? Undershoot the and you'll get to see nice oscillation. mRNA and protein. And again, you will need to follow on those rules of sufficient non-linear. And just one small point. If you look at this particular picture here, which I've shown, initially you see there is a stable steady state. There is this unstable steady state. And again, there is a stable steady state. Right? So this is definitely a bifurcation diagram. Now, in this unstable region, the protein oscillates. I've shown that the proteins and mRNA oscillate when the signal is in this region. Beyond this region, they won't track this region. Okay. So definitely, some crossover is happening at this particular point between a stable and a oscillatory signal. First a point called Hopf bifurcation. We'll not be discussing the details of exactly how a Hopf bifurcation works. For the context of our biological discussions, just understand that whenever a system crosses from a stable to an oscillatory zone, that crossover point called a Hopf bifurcation. If you want to understand the physical significances of such Hopf bifurcations or how they arise, you can follow the book of Shogart Swanning. There is a very good book. But for our, yes, just assume that just some saddle nodes it had changed between a stable and an unstable. But in case of saddle nodes, that unstable was not oscillatory. But in case of Hopf bifurcation, it will be a stable followed by a oscillatory unstable. I think we conclude our discussion up to here at this point. I just want to end with what are these particular sources of nonlinearity. So one which I mentioned in terms of Hill coefficient, that whenever you have a oligomerization process, your timer, primer, tetramer, for the run, each one step will keep on adding one into your Hill coefficient. Right? So that is one source of nonlinearity. One more source of nonlinearity, if you remember, is cooperativity and LSC. Why cooperativity? If you remember in case of hemoglobin, one source of nonlinearity was that it binds in a tetramer process. But if you also remember, it binds 4 oxygen molecules, and each binding actually enhances the binding of the next molecule. So there is a cooperativity in it. So that is another. Also, I mentioned that phosphorylation of the protein and dephosphorylation. So phosphorylation, multi-site phosphorylation. So if you have some amino acid that for one position you can phosphorylate. So each step of phosphorylation will provide one glucose chain. So that is another source of nonlinearity. Also, there can be stoichiometric inhibition. We have not discussed this, but if you remember your enzyme kinetics, whenever you have competitive inhibition and uncompetitive or mixed inhibition, you see that the system stops being a linear function or rather the normal michelation function, and you start getting all these non-linear behaviors. So biology brings in non-linearity by majorly these things. So either oligomerization or either cooperativity. We'll keep our discussion to this point and tomorrow, 3 p.m. So we will meet 3 p.m. tomorrow. And oh, sorry, not from 3 p.m. Tomorrow the class will begin at 11 a.m. and we continue at 11 a.m. till 1 p.m. Thank you.